Welcome back, students. In previous video we have discussed about the pathogenicity, pathogenesis and anthrax cycle of Bacillus anthracis. In this video we will discuss our remaining part, that is lab diagnosis, prevention, prophylaxis, treatment and control. Okay, so lab diagnosis, let's begin with lab diagnosis. The lab diagnosis can be done by the microscopy, culture, animal inoculation, serological test and different techniques are being included. When an animal is susceptible to die because of anthrax, autopsy is not possible. Okay, autopsy is not permissible. Why? As the blood may contaminate the soil. Okay, so it is not possible to do the autopsy means post-mortem. Okay, so it's not possible. So what is option for that? An ear may cut off from the carcass is the word to describe the dead animal, okay, or dead body. So an ear may cut off from this carcass and it may send to the laboratory for diagnosis, or alternative for this is either swab collection is there or blood collection is there. Okay, then it is sent to the laboratory for the diagnosis. Then the demonstration of the gram positive bacilli with the morphology of anthrax bacilli and McFadden's reaction can also be used, and this immunofluorescent microscopy will confirm the identification, because in this technique there is a visibility of the capsule, and capsule is the main antigenic structure to cause the disease. So this technique gives the confirmation of this disease, that is immunofluorescent microscopy. Now different techniques are there. First is sample collection. Sample either blood, CSF, that is cerebral spinal fluid, or sputum or pus can be collected. Once the sample is been collected, it is been micro वो ऑब्जर्व किया जाता है. देन ऑब्जर्वेशन इज बाय माइक्रोस्कोपी. ग्राम स्ट्रेनिंग कैन बी डन. in gram staining, gram positive rod shaped bacilli are been observed, or you can go for the McFadden's reaction. The stain which is used in McFadden's reaction is polychrome methylene blue stain, as we have discussed this reaction in our previous video also in biochemical characteristics. This stain gives the observation in the form of purple capsule surrounding blue bacilli. Okay, so this is all. Or you can go for the spore staining. Once the sample is been collected, it is been observed, then the sample is been culturalized. Okay, कल्चर किया जाता है. तो कल्चर कहाँ करेंगे? ऑन टू द न्यूट्रिएंट agar also. On to the nutrient agar, the colonies are observed in the medusa head like appearance. Okay, you can see the diagram. circular white dark पैच है और उसे सराउंडिंग मेडुसा लाइक स्ट्रक्चर है. मेडुसा लाइक स्ट्रक्चर मीन्स वी कैन से थीन थ्रेड और फाइबर लाइक स्ट्रक्चर है. Next you can also grow this onto the blood agar. Onto the blood agar, it shows non hemolytic property. मतलब there is no there is no rupture of RBCs. Okay, there is no rupture of red blood cells. That is the non hemolytic property. और जो सिलेक्टिव मीडिया है स्पेशली फॉर दिस बैसिलस एंथ्रासिस, इट इज द प्लेट मीडियम विच स्टैंड्स फॉर द पॉलीमाइक्सिन, लाइसोजाइम, EDTA एंड थैलस एसिटेट. So this is the selective media for the Bacillus anthracis. Thallus acetate, it is the salt of thallium and acetate which is used as the selective growth for this Bacillus anthracis. Okay, so this is all about the culture. Next technique is gelatin liquefaction. This also we have discussed in our बायोकेमिकल कैरेक्टरिस्टिक्स. इसमें क्या होता है? लिक्विफिकेशन होता है, मैक्सिमम लिक्विफिकेशन होता है फ्रॉम टॉप टू द बॉटम. ओके, you can see in at the top layer how the liquefaction is been done. और इसका जो appearance, it is like inverted fir tree appearance. Okay, fir tree, the leaves of this tree are very thin, so उसका it has been resembled to the results are been resembled to this fir tree. Okay, so the name is given inverted fir tree. So this is about the lab diagnosis. And other techniques involved in the lab diagnosis are ELISA technique and we can say complement fixation technique. ELISA technique, it stands for the enzyme linked immunosorbent assay. This technique determines the antibody present in the patient serum. And the next technique is complement fixation technique. In this technique, particularly specific antibodies are being detected. Okay, complement fixation technique में specific antibodies against that disease is being detected by using sheep red blood cells, sheep RBCs and antigen antibody. Okay, तो ये कुछ techniques हैं for the diagnosis of Bacillus anthracis. Next is the prophylaxis. Prophylaxis मतलब prevention. Okay, how the disease can be prevented. It is said that prevention is better than cure. Okay, disease होने से पहले ही आप prevention लीजिए ताकि डिजीज होगा ही नहीं. सो आप कैसे प्रिवेंशन ले सकते हैं? प्रिवेंशन डेफिनेटली हाइजीनिक कंडीशन मेंटेन करके, प्रॉपर हाइजीनिक कंडीशन अगर आप मेंटेन करते हैं, स्टेराइल कंडीशंस अगर आप मेंटेन करते हैं, डेफिनेटली डिसीज विल नॉट हैपन एंड रिमेन बी अवे फ्रॉम दी, वी कैन से बी अवे फ्रॉम दी एनिमल्स, पेट एनिमल्स. अगर मतलब उनकी प्रॉपर हाइजीनिक, I'll not say that be away, but maintain, if you are having pet, maintain the good hygienic condition of that animal also. Okay, so that there will be no any disease to that animal. If there is no disease to the animal, ultimately disease transfer होगा ही नहीं. तो आपकी भी अगर आपके पास pet है तो maintain the good hygienic condition of that pet also. एंड इससे पहले प्रिवेंशन के लिए तो वैक्सीनेशन अवेलेबल है ही. ओके, देयर आर वैक्सीनेशंस अवेलेबल फॉर दिस anthracis, which was firstly developed by the Pasteur, which is known as the Stern vaccine. Okay, so Pasteur anthrax vaccine is of great historical importance. तो इस तरीके से आप प्रिवेंशन या फिर प्रोफाइल एक्सेस मेंटेन कर सकते हैं just by maintaining hygienic condition of pet animals plus yourself also. Vaccination कीजिए so that there will be no any anthracis disease. Different vaccines are there: Stern vaccine, Mazuchi vaccine and Carbozzi strain. Carbozzi strain, this is mainly used for the spore staining or for the identification of the spores. Okay, so this is all about the prophylaxis. अब ट्रीटमेंट में हम क्या कर सकते हैं? तो ट्रीटमेंट एज there are two types of diseases caused by the Bacillus anthracis. तो वो दो टाइप्स कौन से हैं? एंथ्रैक्स दो टाइप्स के हैं. तो एंथ्रैक्स फर्स्ट इज द क्यूटेनियस एंथ्रैक्स एंड सेकेंड इज द पल्मोनरी एंथ्रैक्स. एज आई टोल्ड यू अर्लियर, क्यूटेनियस एंथ्रैक्स it occurs greater than 95% of the population. greater than 95% इसका अमाउंट है. ओके, तो ये क्यूटेनियस एंथ्रैक्स एज इट ऑकर्स ऑन टू द स्किन, इट इज रिलेटेड टू द स्किन इन्फेक्शन. इट इज ट्रीटेड बाय द एंटीबायोटिक्स. अब आप ट्रीटमेंट कब लेते हैं? प्रिवेंशन आप डिसीज होने से पहले लेते हैं और ट्रीटमेंट आप डिसीज होने के बाद लेते हैं. ठीक है, तो क्यूटेनियस एंथ्रैक्स इट इज ट्रीटेड बाय एंटीबायोटिक्स लाइक there are three main classes of this antibiotics like tetracycline is there, macrolides is there and fluoroquinols are there. Now in tetracycline, example of antibiotic is doxycycline. Okay, doxycycline ये antibiotic preferable है. Second class is macrolides, in that the antibiotic is erythromycin. Third है fluoroquinols, उसमें antibiotic है ciprofloxacin. So these are different antibiotics which can be used for the treatment of cutaneous anthrax. Second type is pulmonary anthrax. Now in pulmonary anthrax, it is rare type of the disease but it is severe. रेयर है लेकिन सेवियर फॉर्म में होता है. तो सिर्फ एंटीबायोटिक देकर चलेगा नहीं, यू हैव टू टेक द मल्टी ड्रग थेरेपी. ओके, मीन्स एक के साथ आपको 2 या 3 डोज लेने, 2 या 3 और एंटीबायोटिक्स लेने पड़ते. सो इट इज़ द मल्टी ड्रग थेरेपी. तो इसमें कॉम्बिनेशन ऑफ, यू कैन टेक द कॉम्बिनेशन ऑफ सिप्रोफ्लक्सिन, रिफैम्पिन एंड वैनकोमाइसिन. ठीक है, ये 3 ड्रग्स का आप कॉम्बिनेशन ले सकते हैं: ciprofloxacin, rifampin and vancomycin. So the patient which is suffering from this pulmonary anthrax can take the multi drug therapy. Even vaccination is also present, okay, which comes under the prophylaxis or prevention. Penicillin and streptomycin are no longer used for the treatment, ठीक है, यही ड्रग्स, यही एंटीबायोटिक्स यूज होते हैं फॉर द ट्रीटमेंट ऑफ द cutaneous anthrax and pulmonary anthrax. For the cutaneous anthrax, single antibiotic is sufficient, but for the pulmonary anthrax, multi-drug therapy is been used. Okay, now apart from this, there are different types of species like anthracoid bacilli, Bacillus cereus and Bacillus cereus. Okay, ये different types of species है under the Bacillus, but our main focus is Bacillus anthracis. So these are the different we can say different species, but in the syllabus just anthracis has been there, Bacillus anthracis. But let me tell you about this species also. Now this anthracoid bacilli, it is associated with the septicemia, meningitis, pneumonia diseases, and it is the opportunistic pathogen, means जब opportunity आती it turns into the pathogen, okay, like Bacillus subtilis and Bacillus licheniformis. These are also an opportunistic pathogen, as I have told you in the classification कि ये दोनों भी opportunistic pathogen है. So this is about anthracoid bacilli. Next is Bacillus cereus. Now Bacillus cereus ये जो species है, it is related to the gastrointestinal disease, mainly diarrhea. Okay, diarrhea cause करते हैं, इसके symptoms है nausea and vomiting. So just diarrhea is the main causative agent. Diarrhea is the main symptom for this Bacillus cereus. ओके, सो दिस इज ऑल अबाउट द बैसिलस एंथ्रेसिस. हमने अब तक क्या क्या डिस्कस किया इन तीनों वीडियो में? वी हैव डिस्कस्ड मार्फोलॉजी, वी हैव डिस्कस्ड क्लासिफिकेशन, देन लैब डायग्नोसिस, प्रिवेंशन, ट्रीटमेंट, pathogenesis and virulence factor. I hope you might have understood this pathogenic bacteria called as the Bacillus anthracis. Thank you.